talk a little bit about my research, the personalized nutrition project. Now, I had to justify my being here in a big data conference, so I took a look at our data to see what we have. And up till now, we have about 2 million data points of glucose data, one every 5 minutes of more than close to 1,000 people. That's about 17 years of glucose documentation. We have about 50,000 meals documented in our database, which amount to 15 tons of food and beverage, and 11 million calories. And also we have a lot of bacterial information about people, about 2 gigabytes of bacterial information about each person. But first thing first, let's start at giving you some motivation to why is this an interesting problem. So there's a lot of metabolic disease going around in the world today in terms of obesity rates, diabetes, and prediabetes. And it costs a fortune to manage. Now, some of you maybe have seen this. These are the United States obesity rates by state. And this is similar to what's happening in the developed world. And you can see that in the 80s, we started around 10% obesity per state. And this progresses, inventing new categories as we go along, with more and more obese people in the United States. Until in 2010, we have no states with less than 20% obese, and many states with more than 30%. So this is an epidemic. And the prevalence of diabetes has also risen to about 7% of the adult population, with a positive trend. And it costs a fortune. In 2012 alone, diabetes, the cost of diagnosed diabetes in the United States stood on $245 billion. Israeli GDP that year, $257 billion. So that's the same ballpark, and it's a huge burden on healthcare systems around the world. Now, some may say that people get sick because they live longer. But this is not true, because you can see a rise in obesity and diabetes in children as well. Some say it's genetics, but that's also not the case, because you can see that in immigrants, they develop the metabolic diseases common in the country of destination, as happened with Yemenites in the 50s and Ethiopians more recently. Others say that we're just insubordinate. Maybe we don't eat what we're supposed to eat, but in fact we are eating exactly what we're told to, more carbohydrates and less fat. So what is it? If we're not eating right, then what is eating right? So as usual, the US Senate had an answer for that. So in 1977, they formed a committee on nutrition that ended up with this conclusion and this famous food pyramid. The obesity epidemic hasn't started yet, so what they targeted was heart disease. And they recommended a decrease in fat consumption and an increase in carb consumption. But since then, the incidence of diabetes and obesity has risen significantly. Now, this switch to carbohydrates is not a recent thing, especially simple carbohydrates and sugars. What you can see here is the per year consumption of sugar in the UK. And you can see that the annual consumption in 1700 is equal to the daily consumption today. Sugar is now 20% of our diet. And the adverse effects of sugar have all been shown in the 60s. In this research, for example, they took 19 people, put them on a high sugar diet for 2 weeks, and then on a balanced diet for another 2 weeks. And during the high sugar diet, they had worsening of blood metrics, and they gained some weight, and this returned to normal after rebalancing the diet. Also, many studies have shown that diets that cause a high postprandial response, this is the sugar response after a meal. When it's high, then the area under the curve there is high. So diets that cause this response are associated with weight gain, with type 2 diabetes, and with high risk for cardiovascular disease. So what we need, in essence, is a diet that would be easy to follow, maybe a general recommendation that would work for everyone and lower blood glucose, right? Problem is, it's not that simple, because people respond very differently to even the same loaf of white bread. Not only do they respond differently to the same white bread, also the ratio between the response to the white bread and the response to glucose, this is called the glycemic index, is also variable between people. And this is why we started the Personalized Nutrition Project, to find out what's best for each person to eat. We're looking to decipher the major personal component in people's glycemic response to food. Now, we've already looked at more than 900 people and about 50,000 meals, and we have a lot of people waiting to be tested. And this is an overview of our cohort. You can see that we have people in all ages between 18 to 70, more women than men. We're looking mostly at healthy people in terms of hyperglycemia and diabetes. So we have about 70% of healthy people. And in terms of BMI, we have about half of our cohort is within normal weight, and about 30% are slightly overweight, and the rest are overweight to obese, which is very similar to the general population. And this is an overview of 50,000 meals that we have in our database. And you see that most of the meals are sort of what you call balanced, with 30 to 60% calories from carbs and the rest from fat and protein. Now, this is how we get this amazing data. People register to the study online. They go to personalnutrition.org, and you can go as well. You fill out medical and dietary questionnaires, and they're invited to a meeting. In the meeting, we take your blood samples, and we take a genetic sample, and we sample your microbes via a stool sample. Then we connect you to a glucometer that measures your glucose levels every 5 minutes for a duration of the week, and we ask you to log your activities and everything that you eat with our app. At the end of the week, we get great data, and people get to see what their glucose metrics look like, what their bacteria look like, and in the future, they'd be able to plan their personal diet, and we'll get to that later. Now, this is a little, in a little more detail how it looks like. I hope you can see that. This is just a screenshot from our website. People can see their glucose metrics, where they are in terms of with respect to the population, and they can see which foods caused a higher glucose response. Now, people actually use that, and a friend of mine who's a pilot in an airline came to me and said, well, you know, I'm getting really tired around the time of landing. So I told him, okay. I'm not flying in your airline anymore, but maybe I can help you by, you can join my study. I was looking for participants then, so it was an easy thing. So he joined my study, and he found out that whenever he eats white bread, just white bread, anything else is okay, but when he eats white bread, it causes his glucose to rise and then plummet, and 2 hours after he's eaten the bread, he's tired. So he stopped eating white bread while cruising in a cruising altitude, and he's no longer tired when landing, which improved the safety in the Israeli airspace. So no, this is just an anecdote, but it's really useful to some people. And also you can see your food intake during the week, the amount of exercise that you had, your blood tests, and you can take a peek at your bacteria, and I think we're one of the first in the world that give this information completely for free. And I'd like to share with you some exciting preliminary observations from our data. First of all, we see the same huge variability in response to bread and to glucose that we see that we saw before in the limited controlled trial. And the relative response between the two is also variable. We have some people that respond higher to glucose, and some people that respond higher to bread, and some people that respond higher to bread and butter. But how does this translate into action? Imagine that you're going to a dietitian and you ask them what you should eat, a bowl of rice or a cone of ice cream. So most dietitians would say a bowl of rice, and rightfully so. But for this participant, it's better to eat a cone of ice cream than a bowl of rice, and we have many like this one. Or for example, if we compare the response to an apple and to baked sweets for people who had both. Each dot here signifies a person. And you can see that some people have a higher response to baked sweets, and some people have a higher response to apples. And there are many such food in which a general recommendation cannot be given. This is the yellow portion of the matrix. So, for example, when comparing an apple and banana, or when comparing bread and pasta, we can't give a clear answer of what should you eat. So there's no general recommendation due to the great interpersonal variability. But what drives this huge variability? So, first of all, there's genetics. Some people can eat whatever they want and they never get fat and they never get sick. Then there's lifestyle. Some people are couch potatoes and some people are marathon runners, and it's two different metabolic systems altogether. And last but not least, there's our microbiome. And some of you have heard about the microbiome, but I don't know if you've seen it. This is how it looks. The bulges are the cells in our small intestine, and the hair-like structures are the microbes. And you can see the very intimate relationship between us and our microbes. And it's truly amazing because we have more microbes than human cells in our body, and they have a lot more genes than we do. And they have a huge effect on our physiology and our health. And the greatest thing about those microbes is that unlike genetics, which we're born with and we live with forever, we can change our microbiome pretty easily by changing what we eat. So recently, the study of the microbiome has become very easy because DNA sequencing has become so cheap. So I'm going to show you a few research directions taken on the microbiome. So, for example, here you can see that cirrhosis, which is liver disease, can be accurately predicted just by looking at the person's microbes. Each column here is a person, and each row is a microbe. And you can see that people with cirrhosis have a unique signature of microbes compared to healthy people. So we can say that the microbe reflects our health. But it doesn't only reflect our health. In this study, they took germ-free mice, and they transplanted microbes from twins, from an obese twin and a lean twin to the same mice. And after 2 weeks on the same food, the mice that got the obese microbiome got fat, and the mice that got the lean microbiome remained lean. So the microbes don't only reflect our health, they also can radically change it. And this has been shown in humans in the context of type 2 diabetes. If we take the microbiome of a healthy donor and transplant it into a diabetic recipient, there's an improvement in insulin sensitivity, which is a metric of diabetes. And this doesn't happen in the control group. And also in this study from Eran Elinav's lab in collaboration with our group, we've shown that artificial sweeteners such as saccharin can promote diabetes by changing the microbiome. So in fact, substances that were considered inert before are in fact not inert in the context of the gut microbes. And what I'm showing you here is just one way by which we've shown it, that mice that got diabetic when drinking artificial sweeteners were cured after being given antibiotics and killing the gut microbes. Now, this brings us back to the Personalized Nutrition Project. As I said earlier, we want to help people improve their dietary choices by predicting their response to untested foods. And this is the way it's done today. The way it's done today is that, especially in diabetics, they look at the amount of carbohydrates in a meal, and they use it to approximate the glycemic response. And it works, but it doesn't work all the time. And we think that we can do better, and that's why we set up the project. So instead of just looking at correlations, we can try to predict people's response to food. So we feed this model with all kinds of features. For example, meal features such as the weight of carbohydrates we saw before. Logged features such as the time of day during the meal, how much sleep the person had. Microbiome features of all kinds, and personal features such as blood tests and questionnaires. So for each of the meals in our data, we have 200 nutrients, including macronutrients and vitamins and minerals and fatty acids, etc. We have meal times, sleep times, quality of sleep, exercise, stress, whatever people log in their app. And we extract the abundances of all microbes that we can look at in people's guts, and all gene groups that we can find, and a lot of genetic modules, for example, sugar consumption modules that are common in those microbes. On top of that, we have the blood parameters, we have questions in our questionnaire, and we have food frequency questionnaires that we use to get a sense of the larger diet that the person has, not just only in the week that they join our study. And one of the big challenges here is to make sense of the raw data in the microbiome of the bacteria. This is our raw data. This is what it looks like. These are sequencing reads. There are small DNA sequences, usually 50 to 100 characters or base pairs long. They have QC information with them. They're usually paired with some other sequence, and they're barcoded per sample to ease connecting them to a person. And we have 10 million read pairs like this per person. So it's about 2 gigabases of DNA data per person. And I think this is unprecedented, not just in Israel, but in the world in terms of microbiome studies. So we spent the past couple of years in developing tools for turning reads into features. So our pipeline includes all kinds of filtering and QC and quality trimming and filtering of human reads. And then we map the reads to a microbial database to get compositional features and map them to gene databases to get functional features. We assemble them to get whole new genomes and work HMMs on them to find motifs. And we develop all kinds of tools for data exploration, for filtering of features, for transformation, decompositions, and to visualize the data to see what's actually interesting for the predictor. And of course, there's a predictor. So when we look at meal features alone, we're already doing better than what is used today. And when we add in the logged features, which signify a person's lifestyle, we get an improvement. The microbiome gives another improvement. And on top of that, we have the personal features. And I'll just say that this is work in progress, and it improves daily, so we expect it to be even much nicer. And also, if we don't look at, even if we don't try to predict the exact number, yeah? Okay. If we don't try to predict the exact number on the glucometer, we have a much easier job. If we just want to say this is a good meal or this is a bad meal, we have a very high accuracy. So our vision is that people would log what they eat into our app. This is our app. And we get such a prediction, good or bad. You should eat this meal, or you should maybe eat chatzi mana. And this is just around the corner. So this is very preliminary, but trying such a division into good and bad meals on a few pre-diabetic patients for 2 weeks ended up decreasing their overall blood glucose. And if they kept following the good week recommendations, they ended up losing weight and ended up improving their metrics. So we may not be winning the battle against diabetes, but I think this is, I hope I convinced you that this is a good tool in this war. And I'll just want to say that this project is a collaboration of a lot of people, the Segal lab, which I'm part of with Eran Segal, and the Elinav lab, which are experts on the microbiome. We have people who run our experiment in Ichilov, based in Zamir Halperin's gastroenterology unit, and people who run our operation in Weizmann, namely Dr. Didi Israeli. And thank you for listening.